I'm going to start with sort of describing how we test drugs. Drugs don't come out of the air. They're not just given to us by Genentech or Novartis. These are drugs that have to pass certain hurdles in the laboratory. They might be drugs that are specifically designed to attack a certain pathway that we think might be important for cancer. They might be specifically designed to attack a certain molecule that we know is on cancer cells. Or they may be drugs that just by chance looked like they were pretty good in the laboratory at killing cancer cells, and that they were relatively safe in mice and cured a few mouse tumors, and therefore kind of jump forward to be ready for human studies. So we start with preclinical work where in the laboratory they show that a drug can work on mouse tumors. Now, one of the problems for neuroendocrine tumors and one of the problems for the patients is that we don't have good mouse neuroendocrine tumors. We have tons of mouse breast cancers, tons of mouse pancreas cancers, colon cancers. We've had a rough time growing mouse neuroendocrine tumors. There are one or two, but we wonder whether they're even valid. So we need better tumor models. So that's one of the areas of investigation. Once we have demonstrated that a drug looks promising enough to try in patients, then we do what we call a phase 1 trial. Now, there are a lot of misconceptions about phase 1 trials. Phase 1 trials, the scientific endpoint is to establish safety. But establishing safety is not really how it's interpreted. It's defining toxicity. So, of course, if we're given a new drug, we have to make sure that we know, well, what are the side effects? How is it metabolized by the body? How long does it last in the body? Does it have any effects on the kidney or the liver? Does it have any effects on the person in terms of diarrhea, hair loss, numbness, tingling, nausea, any of those side effects that many of you know all too well? But, so the scientific endpoint is just how does the body handle that drug? But that doesn't mean we can't hope for efficacy, because these are drugs, after all, that cured a few mouse tumors. We're hoping that they can have a role in patients too. So we have phase 1 trials where we're testing drugs in people. Because they're phase 1, because the scientific endpoint isn't what percentage of patients with colon cancer have a response, what percentage of patients with neuroendocrine tumors are cured. Those are nice endpoints, but the scientific endpoint is safety and tolerability. And therefore, any patient would be eligible for a phase 1 study. So if you go through the NCI.gov list of clinical trials, you won't find a whole lot of clinical trials for neuroendocrine tumors, but you'll find a lot of phase 1 trials where neuroendocrine tumors would be eligible potentially. And again, the scientific endpoint is safety. Anyone with a measurable tumor is usually anyone who's in good shape with a measurable tumor could be eligible, and you can get a phase 1 drug, or combination of drugs. Phase 1 trials are usually small, 15 to 25 patients or so. And again, it's to define side effects and try to determine what's the best dose of the drug. Well, if the tumor passes the mouse test and is determined to be safe at a reasonable dose in phase 1, the next question is, how effective is it? And then we have to take a guess. We have to say, well, let's test this in colon cancer. Let's test this in breast cancer. Let's test this in lung cancer. Do you know why they test those 3 cancers? It's a big market. I mean, number 1, it's a big market. Number 2, they're common. The fact that they're common means that you can test them quickly, which is not, it's one of the problems is how long it takes to do clinical trials. So you don't get too many drugs tested first in neuroendocrine tumors, although that may be changing soon. So the question then is, how effective is it in a defined population of patients? So you define a cancer. So most phase 2 studies are disease-specific. Okay, phase 2 study is asking the question, how effective is this drug for colon cancer? How effective is this drug for pancreatic neuroendocrine tumor? How effective is this drug for carcinoid tumors? Phase 2 studies are usually limited to 25 to 50 patients or so. And we just get a kind of a ballpark estimate of that. If we think that the 25 or 50 patients here did okay, we think that the drug looks reasonably effective, then the next phase of study is to say, well, how does that compare with the standard treatment? So we've already established safety. We've established that it does work for some patients. So we think it works for enough patients to take it to the next step. And this is the one the FDA likes. It's a phase 3 study. It sometimes gets bad press, but it's a randomized study. It takes patients with the same disease. If it was colon cancer here, it would be colon cancer here. Breast cancer here would be breast cancer here. If it was neuroendocrine tumor here, it would be neuroendocrine tumor here. And the phase 3 study simply asks the question, how does it compare to a standard treatment? Now, it may be that the placebo is a control, which is to say that if you have a disease for which there is no standard treatment, then it's perfectly ethical to randomize between drug X and a placebo. If you have a disease with an accepted standard of care, then oftentimes what we'll do is we'll say, what about the standard of care plus placebo versus the standard of care plus drug X? And then at the end of, well, this study might take 200, 500 patients. You can see that that's a limitation already when you saw the numbers that Dr. Visser was showing you of surgery studies for pancreas, for neuroendocrine tumors with 20 or 30 patients. It could take 200 to 500 patients to do an adequately powered, statistically significant phase 3 study. Now, what I didn't put up here is time. This study might take 6 months. It could take a year and a half. This study, depending on how many centers participate, it could take a year or it could take 3 years. This could take 3 to 10 years to do. You add them all up, it's maybe $20, $30 million. And then ultimately, if this one's positive, the FDA says thumbs up. Okay? So that's the process that we go through in clinical research to try to get a drug approved. I'd be interested in Dan's process for getting procedures approved is much shorter than this, but this is what it is for drugs. Now, there are important things to recognize when we look at studies, because the ultimate outcome of the study depends on the design of the study, and so we have to really be careful with our interpretation. I'm sorry, this kind of got scattered when I went from Mac to PC. So let's think about eligibility criteria, meaning we have to define a population of patients and then decide, well, this is the group we're going to treat. And so the outcome might only reflect that group of patients. But maybe we can extend the interpretation of that outcome to maybe affect other groups of patients that were not actually studied in that trial. But you always have to remember that there's a defined group of patients for which that treatment was offered. And it might not affect you or the person sitting next to you because they might be a slightly different disease or a slightly different population. So we had a nice talk this morning from Terry Longacre talking about the neuroendocrine tumor types with well-differentiated, poorly differentiated. She called them grades, grade 1, grade 2, grade 3. Those are 3 different, biologically different diseases. And well-differentiated can behave very differently from poorly differentiated tumors. In fact, when we talk about neuroendocrine tumors for the purposes of this audience, we don't even include these. That's a tough crowd. The well-differentiated or moderately differentiated or grade 1 and grade 2 are the ones that we usually talk about. We also talk about the site of origin. Do you just combine all neuroendocrine tumors? And because they're so rare, it makes sense we'll just take any comers with neuroendocrine tumors so we can get our 50 patients and publish our results. But is it fair to combine apples, oranges, bananas, all kinds of different fruit, when in fact there may be significant differences among neuroendocrine tumors depending on where they arose, whether they arose in the stomach, the colon, the small intestine, the appendix, the pancreas. One really key part that's becoming more and more important is, was that tumor growing? Which sounds silly. Tumors grow. Some patients with neuroendocrine tumors grow so slowly that it might be years before you could even see it on a scan. And you can follow it on a scan, just follow a millimeter at a time, and I've had patients I've taken care of for 10 years and never treated them. Never treated them with anything. And yes, if I look from one scan to the next every year, there's no change. If I look this year's scan back 10 years ago, it might be this much different. So that's slow growth. Do those people need to have these same treatments? Probably not. But some trials don't stratify based or don't discriminate based on whether the tumor is growing or not. Most of the new ones do. Most of the new ones require that the tumor has demonstrated some degree of growth in order to be eligible for the clinical trial. Prior treatment or not. Some studies will not allow you to be on the trial if you've already been treated. Or some studies will not allow you to be on the trial if you've been treated with a certain type of drug, because they're testing a similar type of drug, and they don't want to compromise the results by accepting people who might already be resistant to that drug or that class of drugs. Okay? So these are really important eligibility criteria that are not always all that well spelled out in certain studies. And part of our work on the NCI task force is to try to get all investigators around the country, including Emily Bergsland at UCSF, who's part of this task force, to accept the same eligibility criteria so that we can start comparing trials between here and Harvard. Measure of success. How do we measure success? It'd be nice to say we measure success in terms of, well, what percentage of patients are cured between drug A and B. Well, cure is a forever word. And it's not quite right that oncologists have trouble saying cure. It just doesn't roll off my tongue as often as I'd like it to. Cure is a forever word. We like remissions. So what's a remission? A remission means that the cancer's disappeared from view. So a complete response is a remission. That's wonderful. We can no longer see the cancer. A complete response that lasts forever is a cure. But that's why we don't talk about cures right up front. We're aiming for remissions. A partial remission means that you've shrunk the tumor quite a bit. And we have academic definitions that Pam might get into a little bit later as to how we define a partial remission. And then stable disease. Well, if this is pancreatic adenocarcinoma and I had a drug that caused stable disease for 6 months, that'd be a blockbuster drug. But if it's pancreatic islet cell tumor and I kept the disease stable for 6 months, the FDA would yawn. They'd say, well, that's no big deal because most of those patients have stable disease for 6 months. So it's highly dependent upon the subset of patients that you pick for the study. If you pick patients with growing disease, then this might be a very important finding. And that's what we're tending to do now. Naturally, if you have progressive disease, you know that that drug didn't work because the disease grew despite it. Well, these are measures of shrinkage or growth of the tumor. That's what response rate means. These are measures of time before the tumor gets worse. Progression-free survival. Progression-free survival means how long does that person survive? That doesn't mean how long the person will live. How long a time will that person go before there's evidence on a scan that the tumor has progressed? Okay? Very simple concept, but hard to measure, particularly for slow-growing tumors. You need a longer time to watch for a slow-growing tumor. Okay? Overall survival. That's an easy one to measure. You're either alive or you're not. And if you're not, that's a data point. If you are, you're a data point. It's binary, right? That's the gold standard. For neuroendocrine tumors, it's very hard to do that because we've got to wait 15 years to do the study. That $20 million study now costs $50 million because I've got to employ data managers longer than my career will last, because you have to study people over a long time if you do that. Now, for pancreatic adenocarcinoma, we can get those results in a matter of just a couple of years because the survival is so poor. So it all depends on the type of tumor that you're measuring.